welcome if you missed the first class. Lab class, if you're in that, meets this afternoon. I didn't get a chance to print the enrollment list, so we aren't going to be taking attendance today, but we're starting out with that rule that's described in your syllabus, and we are just going to roll ahead with that as we go forward. So I made a video of the syllabus as I described, and just 9-minute long video, and so we should start out checking in to see if anybody has any questions about anything at all. So it could be about the expectations for our course, or how do I grade, or the quiz that we have at the end of class today. So I set my alarm at 10:30, and we'll stop then and take this short answer quiz on the first 3 videos that we talked about on Tuesday, and then we'll have another quiz at the end of class next Tuesday, next time we meet. So do you have any questions about anything at all? And then beyond that, we're starting to look at chapter 1. Everybody has access to chapter 1. I think that's our list of things to get done. And even if you don't have the book quite yet, you have access to the first 2 chapters we're going to read, chapter 1 and chapter 7, and on electronic reserve. So that was all described in our first class meeting and on announcements in Brightspace. So there's that electronic reserve pathway. Go to content, which is where you find the syllabus. Also, electronic reserves, then click Aries, and you see our 2 chapters from the book. And that's all I'll ever be able to post there like that. I talked on Tuesday what this course will be like. Here's one more thing about what this course will be like. I help organize a research internship on these topics at Maine Health Institute for Research each school year. And we have funding for about 10 students that are down there each school year. And it's a spectacular research internship. Before I was a professor here, all of my school and training was at gigantic biomedical research universities, which means they had a big hospital on campus, like a hospital that's as big as this campus, all one building, and then all kinds of buildings stuck onto it, and Los Angeles, and Seattle, and Minneapolis, and New York. And so I hung out with, in research labs with pre-med students and med students and stuff like that, all kinds of folks interested in health pathways from healthcare to research and health. And I have helped organize this internship at Maine Health Institute for Research for 15 years, and it was only about 5 years ago that I realized this is the best internship program I've ever seen. And it's amazing, right? Because you're not attending a gigantic biomedical research university that's world famous, nothing like it. But it turns out that this area is just the right size to have a really good research institute of that size, and to build a really nice connection with it. And so I'm happy to talk to students about that anytime. And so the students get paid to be down there. They're down there about 15 hours a week on average. They set up their schedule with their host lab, and they get paid almost $9,000 from USM. That's another thing that I've never heard of before in my time, is that USM pays students to do internships, sometimes on campus, sometimes around in the community. And this one, I think the students get paid more than any other because it got started early. I keep a record. These are students that are going on and they're graduating, right? Going on and doing stuff like that. And so it's a good thing. And it's very similar to the stuff we talked about in our course, okay? So here's where we're at. This is our flow, toward that first exam in about a month, and we'll probably get about that far. So, 1, 7, 16, 17, 18, 19, like that order. And we're starting to think about chapter 1. And chapter 1 introduces cell biology, and I mentioned on Tuesday this is really 2 courses in one, cell biology and molecular biology. And they're kind of, for most people, they're indistinguishable. But for me, with all my background, they are somewhat distinct fields, and it's pretty interesting to think about teaching both of them. In our book, our textbook is broken up into 4 sections. And we're reading 2 chapters from the first section for now, then all of the 4th section, and then we're going to loop back and pick up other sections. So it turns out section 3 is really a cell biology section. So we have chapter 1 is quite focused on cell biology and introducing cell biology, and we won't hear too much more of that kind of cell biology approach until certainly after our first exam and almost after our second exam, because we'll loop back into that and cover some sections like it. But it's a very nice introduction to our course. Why not very much molecular biology in the first chapter? It's because the fourth chapter of our book is all about an introduction to molecular biology. That's the dominant story in this course and in these fields. That's just a huge topic in biology, and it's invaded every other aspect of biology. We're never going to read chapter 4 because we're going to have so much intro to molecular biology through the videos that we're having quizzes on today and next Tuesday, and then also in class, and also when we jump to chapter 7, that's heart and soul of molecular biology. So we'll talk some about cell biology. Here's just a little list of things that I came up with from that kind of chapter in cell biology of the kind of topics that are covered in cell biology. And we're thinking about cells. Cells are the fundamental unit of all life on Earth. And in this course is really, you would say, molecular cellular biology of animals. We really don't talk about plants hardly at all, but we're learning principles that are relevant for plants as well. We also don't talk about microbes hardly at all. So either prokaryotes or single-celled eukaryotes, we don't talk about them hardly at all. Why not? Because there are courses in microbiology, and those microbiology courses are very much cell and molecular biology of microbes like that. Yep. So there are sections like this, and it's nice to look at these, I think, to organize this course, because you're expected to learn from the book, and I won't be covering it all. And when I don't cover it all, it's because I think, the book did a good job explaining that. I don't have enough time to explain the whole book, so I won't explain it. So it doesn't mean if I'm not talking about it, that's not an important topic. So that's one thing to be aware of. Then the next thing is we gotta take this information and distill it. And when I'm not talking about it, you have to do that. And there are two ways to distill coursework, and one is your joy of learning. You're learning about this stuff, and you're getting a kick out of it, and you're fitting into all the other things you've learned and stuff you will learn. And then the other is you're playing a game with me on, what's going to be on the exam. And so it's very important in that second category to recognize that we're going to have these 4 exams, and each one is going to be 50 short answer questions. So that's the context of your learning, is like, what's this, what's that, like that. And this is a good chapter to look into that. Because, for example, in this first section, and I'll talk about it a little bit, but there's really not very much stuff in that first section. And so we have a first section, second section we want to spend some time with, third section, and I'll talk about the third section first, and I'll talk about it for a reason that's important for you to understand also, and that is, often I don't say cell biologists, but cellular molecular biologists are very, very focused on methods. And how do we know that? And there's a lot of that in our book. And when I teach other courses at USM, I'm like, I just really, we don't have enough time. There's so much content, so much stuff that's already known to try to scramble and get into your heads for the first time, that explaining the methods and the protocols is gotta move on. But here, we'll talk about it here, the methods become very looped into the approach and the understanding of what's going on, and the limits of our understanding, like that. And so we'll talk about that a little bit, because the third section talks about some of the tools in cellular biology, like that. So it's a good place to start. And so we'll just talk about 3 methods to kind of guide us toward that, not kind of grinding through the whole, section of that chapter. And here's just a little history, and I mentioned that, it really got going in the 1940s and 50s with the advent of electron microscopes to view eukaryotic cells and say, what's that thing? Well, it's the mitochondria. And in fact, Mitochondria was first identified under a regular light microscope, 100 years before that, but nobody had a clue. And so the two parts of cellular biology that are the root principles of it are seeing that cellular system, and then actually separating components. So we'll talk about that some. So let's isolate the mitochondria, and that'll help us figure out what it does. Or isolate the chloroplast organelles from plants and say, oh my gosh, all of photosynthesis happens inside of chloroplast. And then molecular biologists take over and they say, well, let's break up the chloroplast and look at all the pieces. So cell biology, when somebody mentions that, people usually think, oh, you got to look at the cell. You want to look at the cell. It's primary, fundamental unit of life. And then, especially in eukaryotic cells, it has these organizing areas, these organelles. And then we can isolate those and understand how they're working. And so we'll talk some about that idea. And start out with this electron microscope. So here's an electron micrograph of a cell using a transmission electron microscope, a TEM. And a TEM does what it says. It sends electrons through the sample. The electrons are transmitted through the sample. And so we see an image that's very much like what we see with a light microscope. Light microscopes usually rely on the light being transmitted through the sample. And we see through the sample. And the TEM isn't used very much because it takes quite a bit of prep to prepare the sample, because the molecules in biology don't bend light very well. So the tissues the cells have to be stained in order to really bend the light, most often stained with some kind of metal, like gold, for example. And so TEM. And that's one kind of microscopy, and an example on exam would be, well, what's TEM, or what's not, no, like let's say, describe one kind of electron microscopy. And you could say, well, there's transmission electron microscope, and what's it used for? It's used to look at cells and look at the features of cells through that transmitted light thing. Nowadays, there are really very few applications. Say in the state of Maine right now, I think there are only 3 or 4 TEMs. They're big, they're expensive, it takes a technician or 2 to operate them. And here's an example just to show why would anybody use it. It's a very specialized instrument, and this is a virus called a bacteriophage, and it's a virus that's really, really little, and it would infect bacteria. So bacteriophages are viruses that only infect bacteria, and this is where molecular biologists got their start. So before Watson and Crick discovered the structure of DNA, in the 1940s, the first molecular biologist established that it was DNA that was the heritable molecule, and they used it using this virus. And so the virus has got a really simple organization. The black is proteins, the red is the DNA. This is the bacteria, and it's going to bind, and it does look like a little spaceship and operate mechanically like a little spaceship, and you can see that under a TEM. And it inserts it. And so in the 1940s, Nobel Prize-winning scientists set up this Nobel Prize-winning experiment. They just labeled the DNA with one radioisotope, and so it's probably phosphorus, and they labeled the proteins with another, it was probably nitrogen, and they just asked which one of those gets into the cell, which one of the radioisotopes get into the cell, and it's like, well, the phosphorus. And they're like, well, looks like DNA. And everybody's really surprised because they were just beginning to find out about proteins and the variety of proteins that were out there. And DNA looks pretty drab and homogeneous like that. So early molecular biology, and there are still people that study that bacteriophage. Still molecular biologists making discovery after discovery using that really simple system. And so if you see TEM around, it's probable that they're looking at viruses or something like that. So then here's a very different kind of electron microscope. It's not transmission, it's actually reflected, and it's called a scanning electron microscope, an SEM. And SEMs are used at low magnification, and they're used where the scientist has coated the specimen with gold or a metal, and that will bounce the electrons off, and you can see the structure. And SEMs are pretty common. You can see this, I think probably their most common use is, well, they're used in manufacturing to look at little detailed parts and stuff like that. But here they're looking at the mouth parts of some kind of tick. And it turns out that ticks are often the species identified by their mouth parts. And so, you know, be analyzing these ticks, and we should look at this under an SEM. And so, you know, on the exam, you know, distinguishing like two types of electron microscopy and briefly describe them. Briefly, right? Not a big page of them, but, you know, 2 points. What are they and what are they used for? Like that, right? Okay? So just some electron microscopy. Now, I'll mention that there are a bunch of different light types of light microscopy in that chapter, and I can talk about them, but I think the authors do a good job writing about them. And so, you know, what about this? What about that? And you should read the book and ask questions in class about them as you're looking at them. And so here's the next thing, and that is this idea, and you don't need to see this, but right hand in hand with seeing those under the electron microscope in the 1940s and 50s, the scientists said, well, we got to get a hold of that thing, you know, you're calling mitochondria. So they would break the cells open, and they would separate the parts of the cell. And they did that a lot with centrifugation, where they would just spin the sample. And centrifugation is just increasing the force of gravity so that things sink faster, like that. And they sink based on their specific gravity, density, rather. They sink based on their density, okay? So here's just an example of a tube in a kind of a classic situation. Well, that's interesting. There is some liquid here that's shown on my computer as a kind of a pink, and it's not coming up here. So there'll be some liquid, and it'll be spun, and maybe there'll be a pellet. And the pellet is just stuff that was denser than the liquid, and so it got pelleted at the bottom. And it's little stuff, like a little mitochondria, so you had to spin it pretty hard. It would eventually settle out at room, you know, gravity, but it might take 10 years, you know, it's just not going to settle out very well. And so we have these terms. The liquid after spinning is called a supernatant, and we have a pellet. And so they started out with that. Let's break it up, let's spin it, and try to separate these. And they could get different things separated by changing the density of the liquid, like that, okay? And so here's how they would break them up, famous way to break them up. And this is a thing that our book doesn't mention, but it's just a very accessible tool, method, and it's called a Downs homogenizer. And it's made of these two, oh wow, what's going on with this? So unfortunately our projector is not doing a good job here, because here are the two parts together, and here they're next to each other. And so we already had that issue. And so it looks like we'll have a little frustration with this, and I'll tell the classroom tech people about this after class. But somehow, because I haven't had this issue before, I assume it's not my computer, or maybe it's the connection. But it's the projector in here. That's too bad. And so I can skip over here, and I'll just draw what this looks like. So a Downs homogenizer is two parts. And it's like a mortar and a pestle. They're glass, and they're clear glass, and what you would call the mortar looks like this. It just looks like a test tube with a bell shape up here, so it can hold on to a lot of liquid. Because what you're going to do is you're going to put the pestle in. The pestle looks like this with a little ball on the end, and you're just going to plunge it up and down with your hand, like that. And what's going on is when the little bell shape is in here, there's just a little tiny gap between that bulb and the wall of the test tube. And it's very carefully created so that gap is narrower than the diameter of the cell and wider than the diameter of an organelle. And we'll focus on the nucleus because that's where the DNA is, and we'll talk about that a lot. So we could isolate nuclei using a Downs homogenizer. Just get the cells out, just go like that a few times, and then spin them. And the cell has been broken open. All the other organelles are too small to settle out quickly. The nucleus is big, it'll be the first thing to pellet. So we just spin the cells hard for a short length of time, and we've isolated the nuclei separate. And there are a lot of reasons why we'd want to do that. One is to check. where's the DNA? And we could check it in the supernatant and the pellet and all the DNAs in the pellet, except for the mitochondrial DNA, because there's some mitochondrial DNA like that, right? So it's a separation technology, and that's a very big concept in molecular biology as well as cell biology. In biochemistry, one of the probably two big principles of biochemistry over history is let's separate these molecules. Here it's cell biologists, let's separate these organelles. So we're going to isolate nuclei like that. And so that's a Downs homogenizer, and you would do that, get some cells, and then spin them, and then collect the supernatant and the pellet, right, like that. So let's talk about a practical example. We talked before on Tuesday about when we get to chapter 16 very soon, we're going to learn about FOXO. FOXO is a transcription factor. Transcription factors need to be in the nucleus to do their job because transcription factors regulate transcription, and transcription occurs on the DNA in the nucleus. And so this protein is regulated, as we learned, by excluding it from the nucleus. Excluding it from the nucleus because it gets phosphorylated, and we can just check that distribution, and it's called the subcellular distribution of FOXO. Just take it, and you can either have the growth factor here or not. And when the growth factor is there, and we grind up the cells with a Downs homogenizer, we don't detect any FOXO in the pellet of nuclei. We see it all in the supernatant, like that, right? And then on the other hand, the absence of the growth factor, Downs homogenize it, spin it, and all the FOXO is in the pellet, like that. So lots and lots of experiments like that, using an electron microscope to see things that are really interesting, and then isolating those different components, those organelles, and by centrifugation, and then analyzing them. See what's here, what's there, how is it changing under different conditions like that. In addition to that, just flat out centrifugation, there are other kinds of centrifugation, right? Because this was begun a long time ago, and it was a valuable method, and so there are more, and our book describes them. And so there's something called ultracentrifugation, and that's when you spin really hard, right? Because we could spin so hard that we spin the smaller components of the cell out. So we could spin out the nuclei, save the supernatant, and then spin that really hard in an ultracentrifuge, and we could spin out the ribosomes like that and see that there's messenger RNA attached to the ribosomes and be like, okay, we're getting somewhere now, like that. So it's an ultracentrifuge, and there are different kinds of ultracentrifugation techniques, and our book just rolls through them. And again, it's a short list, but make a list, make a set of flashcards saying, this kind of ultracentrifugation, and this is what it's used for, like that. So let's look at one of those. And I just show you this now because historically, the ultracentrifuges were these gigantic floor-standing units. And in fact, USM still has one of these. It hasn't been used since before I got here. But it's just marvelous to see them because the technology used to have to be bigger. You used to have to isolate more nuclei or more ribosomes like that because the techniques were not as fancy as they are today. And so here's just a comparison of, this is called the rotor where the test tubes go in and it spins. And here's a rotor for ultracentrifuge around that's called an airfuge. And you can see it has the set of parts that's going to spin, has these little fins. And all that's done there is a jet of air is shot at that and it's causing them to spin. That's why it's called an airfuge. And the jet of air is so focused and so fast that this will spin 100,000 revolutions per minute, like that, and just spin a very small sample. So, the technology has changed, but some of that strategy is just, still there, like that. And the most kind of famous thing for an airfuge application is that you have just molecules like maybe an antibody protein or maybe some hemoglobin or insulin, and you want to just get the other stuff out of the sample. You just want to pellet any other bigger materials, kind of garbage, debris type things. So you just spin it hard in there, and it would take forever to spin a protein out of solution, but anything smaller than that, like that, could just be spun out like that, okay? So here's an example I'll just talk about. That's the name of a thing that we can ask about, and it's called equilibrium density gradient ultracentrifugation. And because it's just really beautiful, right? That Downs homogenizer is a really satisfying thing to see. You know, you just go like this and you're done. You know, you've broken the cells up and you've broken things up that were too big to fit between the pestle bell and the walls of the glass test tube. Something I didn't mention is that there'll be different plungers with different bell sizes, you know, so you could, you know, change the size, right? Break open the cell, then isolate nuclei, break open the nuclei with a bigger bell plunger, like that, right? So very satisfying, and that's one reason why we should talk about the methods in our course. It's because you're just like, oh man, great, you know? It's just like, you know, I could have thought of that, you know? Right, like that. So this is like that, okay? And so this is where you're gonna say, we're just not gonna pellet things, because we're gonna pellet, if we did, with this experiment, we would pellet a mixture of things. So instead what we're gonna do is we're gonna make layers of the solution, and the layers are gonna be different densities. And what's gonna happen is we'll load our sample up here, or mix it in, that's what's shown here, and then just spin that really hard, and the samples will float at the interfaces between these different layers, like that. And then, this is a plastic tube, you can use a needle and suck that out, like that. Or pull it all out and check the fractions to see where they separated, like that. And so, this is showing that. Again, our you know, picture is a little bit dissatisfying, because what it's showing here is actually some blue molecules and some white molecules. And the blue, you know, banded at this density, and the white molecules banded up here, right? So we take a mixture of molecules and we can separate them based on their different densities. And you won't be able to see this in detail, but DNA, RNA, proteins, chloroplasts, viruses, skeletal muscle, they have slightly different densities. So we can float them by creating these gradients or layers of different densities in the solution. And that's done in a really surprising way. So you know you could make a salt solution, just take some table salt and some sodium chloride, and when you add the salt, it's going to increase the density of that solution. Turns out that sodium chloride is not very good for creating these stacks or layers of different densities, but cesium chloride, so just another salt, but a heavier salt, is really good at making those. And so there are a couple of just simple salt solutions that can be used to create the solutions needed to stack these. And in some experiments, they're stacked. In other experiments, they're actually just like mixed in as you go, and we have kind of a continuous gradient like that. And so that's equilibrium density gradient. So a density gradient like that, and equilibrium. You spin it till this comes to equilibrium. How long? A couple of days at 60,000 revolutions per minute, you know, like that. It's still the same principle of centrifugation, that things are going to move based on their density, like that, okay? So just a little bit of that, and just some stuff from that third section to see that it's satisfying. And sometimes, you know, you might have a question and you might just Google it and answer it, you know, yourself, because Wikipedia, you know, the first paragraph of Wikipedia for any of this stuff is great. It's written just to target, you know, college student level learning, like that. Or bring them to class, because if you're confused or uncertain about something, you know, everybody else will be also, like that, right? So that's just kind of rolling into a little piece of our required reading to help you see some of that. And that's what biology is like. That's why we have a textbook instead of just having me blab, because biologists have to deal with too much information, and they have to organize their learning in a hierarchical manner. You know, what's the most important stuff here? And our book I mentioned last time has section headings and detailed bold words and stuff like that, and so use those tools and learn about, navigating through, and then ask questions, and I remember that one of the questions is just, what do we need to know after off this page? And I'll say nothing. That's a super satisfying question, right? Because some of the pages have information that's really overwhelming, just a bunch of stuff. And I'll be like, yeah, no, it's just not worth it. We don't need to go there. Yeah. So I always try to bring my book, in case somebody says, what about page, 58, like that, okay? So here's just a little bit, other stuff, right? So light microscopes are limited in their resolution. Their book talks about that. It's based in part on their numerical aperture, and so there's some terms there. And again, I'm not going to try to make you learn everything. We have a lot of stuff to learn, and there won't be a lot of points on the exam that are just from the book. By far and away, the most important thing to do is focus on class, like that, So here I mentioned equilibrium, density, gradient, centrifugation. That's one you better learn. But what about other types of centrifugation? Can you stretch your learning into some more of what the book is explaining, like that, right? So it's called subcellular fractionation. The book talks about it, like that, and so it's good stuff. Then in talking about light microscopes, it turns out that, not that much use for electron microscopes. I always remember when I went out to college and I ran into molecular biology, I went home to my hometown, which was a little farming town in Minnesota, and on break, and I'd tell my high school buddies, I want to be a molecular biologist, and they always would just be like, "Oh man, that's a lot. I guess you're going to be using a microscope all the time." And the answer is yeah, but not an electron microscope. It turns out light microscopes are just the right magnification capacity to look at lots and lots of stuff in light. And so there are a variety of different light microscopes, and we could go through and talk about them. And I'm happy to talk about individual examples, but I think the book does a good job organizing these examples and giving you 2-point descriptions, of each one, like that. So then also some methods. So some methods that use the microscopes. And here's an example called FRET, fluorescence resonance energy transfer. FRET is something that one of the scientists down at Maine Health Institute for Research uses a lot. His name is Igor Prodosky, and right now his intern from USM is called Ashton Dodge. And Ashton's doing a great job, and Igor says, Ashton, you've been working here for a semester, you're gonna work here another semester, can you stay here for the summer? And the answer is yeah. So we also have funding for students that are having a good experience and their host lab thinks they're having a good experience to just keep staying down there. Because again, it's the best internship. So fluorescence resonance energy transfer. The book, I think, does a good job describing it, FRET. And that could be like the hardest question on the exam is, well, what's FRET? How's FRET work? Because it is, a thing you could explain, but if you read it, you'll be like, I have to think about that. And I'm not describing it in class, right? So, these are examples where I'm mentioning, I'm happy to explain it, but take a look at the book. If you have questions, bring those in, because we're kind of rolling forward. Okay. So then here's just a little bit of molecular biology, because we're talking about cell biology, and we'll keep talking about chapter 1. But I talked about those methods a little bit, and before we go on to other parts of the first chapter, just want to mention a couple of things. And one is that the quiz that we have next Tuesday, that's just a bunch of molecular biology. That's just classic molecular biology. And we'll refer to those videos almost every class meeting as we go forward. They're a big deal. And our quiz today sets that up, just kind of frames that. But those are very important videos. And where our big focus is the gene and how the gene functions and the transcription factors like FOXO that bind to the regulatory region of the gene to regulate gene expression. And so, big intro to molecular biology in those videos. And there's just a screenshot of the videos. And here's just an image from chapter 4. And we're not going to read chapter 4. You don't have to look at chapter 4. We're not going to read it anytime this semester. But you're welcome to read it going forward, because it's got a good intro to molecular biology, and so it would help you understand those videos. But again, I've never looked at it, but I'm sharing it with you because you can if you want to, like that. that's where that kind of intro setup in our book is for the videos. That's the intro, and then after chapter 1 we go to chapter 7, and chapter 7 is all about those videos. The quiz next Tuesday, it's transcription regulation in eukaryotes. So it's just blah, 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 and more. You know, it starts with what we learned there, and it just jumps on, you know, further like that, okay? So some of that. Then here's just a cute thing from molecular biology. And it started out as the biochemistry of DNA, so in the 40s and 50s and 60s and so forth. It was, you know, what's DNA? How's it organized? How does it function as the, you know, heritable molecule? And then in the 1960s, we grew into this thing they call the central dogma, and that's the idea that DNA makes RNA, makes proteins. And that's how people say it, and you know that it's not DNA doesn't make it, it's that DNA gets transcribed into RNA, and then RNA gets translated into protein like that. And that's just a bunch of molecular biology like that. And I mention that here for a couple of reasons. And one is that's how the field of molecular biology advanced. So in the 1960s, molecular biologists figured out the genetic code, so the triplet letter, you know, nucleotides that code for each of the amino acid like that, you know, so they were really figuring out the fundamentals. And in the 1960s, the United States was really focused on this Apollo mission. Let's put a person on the moon, you know, like that. And that succeeded in the 1960s. So it was a big, you know, technological, you know, push. And it was Cold War push against the Russians like that, you know, it was a big, big deal. And it succeeded. And so I think it was like 1971, so, you know, 55 years ago, the government said, we're gonna have a war on cancer. And that's where this really takes off, because this is subtitled, you know, molecular mechanisms of cancer, and it starts there. And we want to go there because it's very helpful to see some of that early stuff to understand just exactly how molecular biologists learned about cancer biology. And who did? Well, our author was your age right then. And he, I think, went to graduate school in Miami, Florida, and then he went to do a postdoctoral fellowship in Madison, Wisconsin, and then he moved to Harvard, got a job there. Then he moved to Boston University, where he was for his whole career. And he's working hard on this all the time. So one of the pioneers in the study of molecular mechanisms of cancer. And so, you know, it's just, you know, the 1940s, we got to focus on DNA. 1950s, structure of DNA. 1960s, here's how DNA is organized and how it gets used, like that, yeah? And so lots of molecular biology back then that just roars on. So I put a picture of him in here, and again, I'll mention some people, you know, but that's not because you need to know their name. You never need to know their name or the years. It's just a kind of way, you know, to go there. Like if you have a friend who really likes the Patriots, you know, that's who you think of when you hear anything about the Patriots. You're like, oh, Janet, oh, this reminds me of Janet, you know, Janet knows all about the Patriots. Well, that guy knows all about this protein that's called RAS, R-A-S. And RAS is an oncogene. So we all have RAS in our body right now in the same way we have a hemoglobin gene and an insulin gene. We have a RAS gene. We inherited one from our mom. We have one from our dad. We have some different alleles of RAS, and we'll focus in on RAS because RAS is doing its normal job now, and its normal job is to help cells respond to growth factors and help those cells go through the cell cycle. And RAS gets mutated and causes trouble because cancer, a lot of the story of cancer is cells that are going through the cell cycle when they should not. And RAS is mutated in about a third of all human cancers. It's the most common gene to be mutated in human cancer. Human cancers are caused by multiple mutations. People refer to cancer as a multi-step or multi-hit process. There'll be a mutation, starts causing some trouble, starts growing, and then another mutation, you know, maybe it's a totally different story, you know, but makes the cancer stronger or healthier, you know, grow faster like that. And so RAS is called an oncogene, and oncogenes have the potential to cause cancer. Onco means study of cancer, so oncology is the medicine of cancer, and oncologist is a cancer doctor, like that. So our author, when did he study it? in Wisconsin. And he discovered it in a virus in Wisconsin, because they were studying viruses, because they were molecular biologists, and it wasn't in a bacteriophage, it wasn't a bacterial virus, it was in a virus that infected mice. And when it infected mice, it caused cancer immediately. And so why was it causing cancer? And he and other scientists elsewhere discovered it was causing cancer because it had accidentally picked up a mutant mouse RAS gene. And so that was the foot in the door. The war on cancer, foot in the door in the 1970s is that. There are a couple dozen viruses, none from humans, but from other animals like mouse, rat, cat, monkey, and chicken, like that, of these viruses that have been identified over the years that cause cancer immediately. Now, we know that, say, for example, HIV can lead to cancer, but HIV doesn't directly cause cancer. These viruses caused cancer directly. And in every case in the 1970s, the scientists discovered it was because the virus had a mutation, had a mistake, and it had accidentally picked up one of the host genes, and it was a defective version of it. And so when that went in, it was the defective host gene that caused cancer. And we'll talk about this because it's hard for students to straighten this out in their heads, but our author of our book was in Wisconsin when he found the RAS gene in mice. And then he moved to Boston and Harvard, and doing so, he left viruses behind, and he looked at real tumors in mice, ground them up, put the DNA into normal mice, found that some of them got cancer, identified the gene, and it was RAS. So, Wisconsin, foot in the door with the virus. Harvard, no viruses, nothing like that. But it was the same gene he discovered, and that was really significant right away, because you say, oh, I see two different ways that RAS leads to cancer, either from that virus issue in Wisconsin or this actual natural tumor in mice. And that's a story for all of these, couple dozen oncogenes, and they're really famous. And they all have three-letter names like RAS or MIC or SART, like that. And you'll see that they are famous today. We know so much about RAS because it's been being studied for about 55 years, like that, okay? All right, so just hardcore molecular biology, right? Huge amounts of grants, right, for studying cancer, but then those told them so much about normal health, right? Because they learned so much about these genes and other genes and how they normally work, and also how they can contribute to other diseases, like maybe Alzheimer's, totally unrelated to cancer because it's all about human health. And, they didn't study them in humans, they studied them in other organisms first, like that, figuring them out, get them going, and then move on. So huge amounts of grant money, and that war on cancer is still occurring. And of course, the war on cancer also included, clinical things like different kinds of ways to do surgery or radiation therapy. And we'll be talking about let's look at the genes. And once we find the genes and we find molecular mechanisms of cancer, can we find a drug that could affect that pathway? So we'll get to that. That's chapter 19 in our course. So in molecular biology, we just want to kind of share in that context some features of molecular biology and what it'll be like when we're talking molecular biology in our course throughout the semester. So the first one is, this is just all very, very physical. The RAS gene was in that virus, and nowadays, in our public discussions, we can agree to disagree. Yeah, no, like that, and it just roars on. And I think for, today's student, it's kind of disarming to hear somebody just go, no, we don't need to slow down and talk about maybe this or maybe that, The scientists molecular biologists are the most skeptical scientists. The original molecular biologists were mostly trained as physicists, and they thought biology was stupid and lame, and they were going to turn it into a science, and they were mean. Like Watson and Crick. Well, Crick was a nice guy. Watson, just a mean jerk, like that, and very, very dogmatic, like that. And so, just like, hey, what? Maybe RAS isn't in the virus. And they're like, that's stupid. Get out. There was this guy, Max Delbrook in Caltech, one of these people, and he would come into a seminar, and these were very popular seminars, right? Everybody would want to go. The news media would be there, and he would come in, and there would be some student in there. He'd just be, get out, you won't understand this. Just mean, like that, right? And so super skeptical, just very aggressively skeptical. Side note is this truth, that the National Science Foundation sends me some grants to review once in a while, and they only send them to me. I'm super nice. I was trained to be mean, but I'm super nice. They only send me grants to review because they've never approved of any of the grants they've sent me. So if they want to reject a grant, they're saying, well, send it to Dave Champlin. Because I can just go, oh, there's a lot of reasons why this is no good. Yeah, like that. So physical like farming and car mechanics all the way, like that, So then, next one, it's this reductionism. And this is, the whole is never greater than the sum of its parts. And you're like, yeah, that's wrong. And, again, very dogmatic. We're just going to take it apart. Your brain, nah, it's nothing special. It's just a bunch of parts, like that. And what an attitude. And, we could argue that, like that, but it's that attitude. We're going to break it down, take it apart. How's a bike work? Just take it apart. And find each little part, each little atom and molecule. And next one is a couple done together. So they really focus on model organisms, talk about that, and the research on these model organisms is done in labs, like that. And so here's some examples of model organisms that are the focus of research, and that's an example of how molecular biology has just overwhelmed all of biology. There's lots of focus in modern biology on model organisms. If we can learn about the mouse, we got a pretty good understanding of all mammals. There's some differences in mammals from mice, but it's cool to learn about differences once we know all the similarities, like that. What's going on here in this model organism approach is a couple of things, and one is in a lab. So we're going to just grow them in a controlled environment in a lab and just make this very, very kind of formalized, reproducible approach, like that. Another thing is they're all related. So they're all related, and evolution led to products of life on Earth that are both similar and different. And evolution describes both the similarities and differences. Evolution is like a two-sided coin. You're going to look at one side, it's all the similarities of life on Earth. The other side, all the differences, like that. And so the model organism approach attacks that. We're going to have a quiz in a couple of minutes, and one of the terms is orthologs. These are model organisms because they have orthologs. They're closely related to us. And I mentioned this is that Saccharomyces cerevisiae that causes red to rise. And the fourth chapter we're going to talk about is the cell cycle. You learned about some of that in Introbio because you learned about most of this in Introbio. And the cell cycle, molecular cellular biologists, geneticists that figured it out were studying that species, And that's because they're orthologs of cell cycle regulators of the eukaryotic cell cycle in every eukaryotic cell on our planet. And they're super easy to identify as being the ortholog, just based on the amino acid similarity between those proteins. Like that, where you would say, could we agree to disagree? And you'd like, not in this universe. There's no room for disagreement that's the ortholog, like that. Yeah, it's just remarkable. And so it's this model organism approach, and then into labs. And I just had to put a little plug in for the other big class I'm teaching this semester. It's called, I'll call it ecological developmental biology. And what that's about is this, and it's just fascinating, and that is, now that we know so much about these model organisms, we can take what we know about those and we could pick a different organism, like an unusual mammal, and we can go out in the environment. And so we can look at really complicated life in detail in the environment now, and how the environment helps create the phenotype. So you learned in genetics and Introbio, well, the genetics leads to the phenotype. Really, typically, it's the genetics and the environment that leads to the phenotype in really complicated ways. These people set the stage for that by looking at just one example species really carefully in a lab under controlled environmental conditions. Keep it really simple. We're just going to roar down that pathway in this reductionistic approach. And then we'll know so much that you can go out in the environment and say, hey, what about this BPA in plastic? What trouble is that causing? Molecular biologists will be like, well, I can tell you right now what we should check, like that. And that's true also in a lot of cancer biology. So much has been learned about the way that the cell normally signals and regulates growth and death and things like that, here's a new issue in cancer biology, and you're like, wow, we already know a lot about the organism. It's not a black box at all. We can dive right into this. So we're going to learn about that from that perspective. Here's this from our quiz in just a few minutes, right? This concept of, well, we're never going to see this ancestral gene. But what's going on here is you're used to hearing about organisms that are related in evolution. So, we have some evolutionary relationship with dogs, right? This is at the gene level. So the hemoglobin genes have evolutionary relationship between each other, like that. And if they're in closely related organisms, the hemoglobin genes are closely related, like that. But we just drop down to that DNA level, and we'll learn some in our course about the reading of the DNA sequences, so the inherited information we have, and there are DNA sequencing methods that we'll learn about that can read the DNA letters really fast. And there's just, in human history, one of the most kind of startling combinations in the history of humanity is the emergence of computers and the ability to store and analyze lots of data with the emergence of understanding that the primary heritable molecule of all life on Earth is DNA. Because DNA is linear information, and computers love to store linear information and compare them. And that DNA sequencing was not done at all before 1960. There was nobody that thought we should. And it would start very, very slowly, like that. In the whole world, just a few letters of DNA were read in the 1970s, say like that. And up and up and up, and now it's just trillions and trillions, like that. And I was thinking about it, and I think, you could say something like this, all the other fields of science, they've really zoomed ahead with, computers too, right? We know a lot more chemistry and physics and so forth like that. We certainly know a lot more biology. But I would say that this is probably a fair thing to say, argue this, kind of opinion. But if you go back to, say, everything that was known in chemistry 50 years ago, that was a ton. And I think that what was known in chemistry 50 years ago is not as much as what's known about just DNA today. Just to kind of give you a feeling for just the scale of discovery by just reading it. And all of it points to evolutionary relationships. And I mentioned this on Tuesday, that evolution is a process and a pattern or product. It occurs, and then there's the product. And we and everything about life on Earth appears to be the product of evolution over long periods of time. And nobody could dispute that. That's not a thing where you could disagree upon. When people say, I don't believe in evolution, they're talking about the process, and they're skeptical about the hundreds of millions of years of evolutionary change in the history of life on Earth. Scientists are actually especially focused on the product, the pattern, because scientists, when they collect data, the only thing a scientist ever does with data is they look for patterns. And there are these patterns in DNA that just go on, and they all point to common ancestry over hundreds of million years, like that. That's the pattern, the product, the beauty of life, the facts of life, and the amount of it is just massive. So we're going to have our quiz in just a minute. We're doing great, a couple more minutes. And so here's an example of a model organism, just to show you. It's one you might be surprised to see. This is the embryo of a vertebrate fish called a zebrafish that you could buy down at the pet store. It just looks like a minnow with horizontal stripes on it, and it's become a big model organism. And here's an example of it being used as a model organism. So it turns out that the reason it's a model organism is because zebrafish have external fertilization. The male releases the sperm, the female the eggs, to the outside environment. That doesn't happen in mammals. We're hard to study as mammals because fertilization and development of the embryo and fetus happens internally. That's hard to deal with. This is all external. And normally when you see an egg, like out in the environment, like frog egg in the swamp and stuff, the egg is opaque. The egg in zebrafish is clear, and the embryo is clear. So this is just a regular microscope, and you're going to see right through a living zebrafish. And this one is a transgenic zebrafish where they've modified its genome to include an additional gene that will glow. And it glows in the nervous system cells. So it tags neurons in the central nervous system. And so just an example of a model organism, and once you get rolling on model organisms, it starts out as, they're orthologs, and it starts out as they're easy to raise in lab, and then it becomes, we have lots and lots of stuff we can do to these model organisms. We can manipulate them in lots and lots of different ways. Yep. Okay. And I mentioned this briefly in the videos for next Tuesday's quiz. I say, here's a mouse that glows in its muscles. And we'll learn about how you have to, manipulate the myogenin gene or the MyoD gene in order to produce a mouse that has muscles that glow like that, okay? And so part of the reason I showed that is there's another feature of molecular biology that's a curiosity, I think, and people don't really mention it, and that is when scientists make new discoveries, they turn them into tools for further discovery. And so that's an example there. Where a scientist was studying a jellyfish, just a little tiny jellyfish that lives outside of Seattle in Puget Sound, that glows. And they're like, how does that thing glow? And they figured out how it glows, and another scientist said, oh my God, I could take that glow gene, I could put it into the zebrafish, make the zebrafish glow, and that would be valuable. For example, we could make a FOXO gene where the FOXO protein would glow. And then we could follow it, right? We could do that, the nuclear prep, and we'd say, is the nucleus glowing? Or is the cytoplasm, the supernatant glowing, right? Is the pellet nucleus glowing, or is the cytoplasm supernatant glowing? So it becomes just a powerful tool in further research discovery. And just to mention that, the people who discovered that idea of using the tool, and this is actually quite common, won a Nobel Prize just for the idea of the tool. Like I won a Nobel Prize for just thinking of the PCR method. These people won a Nobel Prize for just thinking about how you could make glowing tagged organisms like that. Another scientist won a Nobel Prize for just thinking and doing some experiments to be able to manipulate the immune system to make monoclonal antibodies like that. So many, many really significant discoveries because they become tools for future discoveries like that. Okay, we got to stop here. So you got to turn off your cell phone.